communicable disease related, and uh, and you can see here the second one is COVID-19. And in that year, interestingly, 8 million people were reported to have died due to COVID-19. But then if you look at um, the SARS-CoV-2 uh, official reported death attributed to SARS-CoV-2 by WHO, the total to date is 7.1 million. Again, showing that some discrepancies, but also showing that tracking such attributing the record of death is not a precise activity, and some estimates of death due to SARS-CoV-2 went as high as 19 million to 36 million. And this is a reminder that really infectious disease, even though often it's associated with low and middle income countries, can and will still affect high income countries, especially in a situation such as a global pandemic. So these pandemics caused by infectious agents called pathogens. So they're organisms that can cause diseases to the host. And they're actually taxonomically very diverse. They belong to different types of organisms. And the genome of these pathogens are all the genetic materials within the pathogenic organism. And these are essentially the blueprint of how the pathogens are built and how they function to cause diseases, and of course, how they function generally. So pathogen genomes are much smaller and less complex than the human genomes, and therefore easier to sequence or to obtain. And this map here shows that, as we are increasingly connected globally, the pathogens can spread with their host much more rapidly, and therefore, again, highlighting why infectious disease need to be considered a global issue and concerns all the nations globally rather than just attribute to certain regions. And I found this map from one of the journals about 2 years ago, and it shows that in the ancient time, the disease are often transmitted just locally, as shown in these green dots. Uh, these are diseases that occurred in the second, third, and sixth centuries. And then the red dot shows the transatlantic slave trade carried diseases from the European colonies to the to North America. And then there's also post-Columbus contact of European colonization, again showing certain diseases such as smallpox and measles travel from um, Europe to the Caribbeans. And then contrasting all those with the purple dots, which is the original SARS epidemic in 2022 to 2024. And you can see here it spread around the globe much more quickly. And of course, this is attributed to the air travels that happen regularly these days. So during the pandemic, the sequencing of SARS-CoV-2 genomes really changed how we conduct public health and how we conduct uh, uh, research to combat infectious disease. So these genomic data allow us to track transmissions and outbreak both locally and globally, to identify genetic variants and use these genetic variants as knowledge to develop clinical tests and vaccines. And also through modeling and phylogenetic analysis, understand how these pathogens evolve and tracing back to the potential origins of these pathogens. And it's already a norm in terms of the research community's mode of sharing pathogen genomic sequence data. And the norm is that the data is often deposited in public and mostly open repositories represented by the National Library of Medicine's NCBI databases. And globally, this database is synchronized with one database in Japan, one in EU, collectively called the INSDC, the International Archive. And during the pandemic, for example, we created a Canadian portal hosted by McGill and administered by the Ontario Institute for Cancer Research, uh, and deposit all the SARS-CoV-2 genomes that were sequenced in Canada by both our public health system and the research groups, and make them available publicly. And there's also corresponding international repositories showing over 16 million, and by now actually more than that, about 17, 18 million genomes available through these public repositories. So there are millions of genomes available out there. And why do we need so many genomes? So if we compare the pathogen genomes to human genome, humans share 99.9% of the genomic sequences and roughly 97% of the genes with each other. If you compare that to two strains of E. coli, they only share less than 50% of the genes, and only 4% of all the genes found in any E. coli are found in all E. coli. So, in other words, they have a big pangenome, and any given E. coli only has a fraction of those genes. If you look at Pseudomonas aeruginosa, a major opportunistic pathogen that's associated with CF patients and other chronic lung infections, less than 1% of all the genes found in Pseudomonas aeruginosa are actually shared by all members of this particular pathogen. So if we need to sequence millions of human genomes to understand human diversity, and by now there's actually about 2 million human genomes partially or fully sequenced, and yet we still don't fully understand how the human genome functions. Imagine how many bacterial genomes we need to sequence in order to understand the microbial diversity associated with these bacterial pathogens. And also, if you look at these public repositories, there are significant amount of missing contextual data. In other words, information about these sequences. So this study here highlighted that in the Gazette databases, 63% of the sequences are missing demographic information. In other words, information about the host. And 84% of the sequences miss the sampling strategy. In other words, missing epidemiological information on how the samples were uh, uh, collected and what the surveillance strategy uh, was. And 95% of the sequences missing patient level clinical information. So we cannot associate the, the viral mutations to clinical outcomes. So in my group, we also conducted a study looking in the literature and looked at over 400 papers, and essentially come to the same conclusion that while in the papers themselves, the information are reported. So for example, sample collection information, the host demographic information, and the information associated with how the sequencing were done, or by and large reported in the literature, they don't always translate readily into the databases. So in other words, manual extraction of the information often is needed. And of course, now with large language models, this process can get easier, but nevertheless highlight a discrepancy between the amount of information that's human readable versus the amount of information that that's machine readable and can be readily extracted from databases. And the reason I title my talk uh, on One Health rather than just talking about human diseases is because uh, over 60% of the emerging infectious diseases are zoonotic diseases, and this includes SARS-CoV-2, um, Zika viruses, avian influenza, Ebola, HIV, uh, and many other familiar diseases that uh, are either highly um, has the potential of causing uh, pandemics or are like Ebola highly dangerous. So therefore, if we only conduct surveillance within the human population, we may very well be missing the next uh, pathogen that would spill over into the human population. So genomic epidemiology in One Health, recognizing that the human, animal, and environment health are interconnected, and genomic sequence data can be used to bridge these different sampling types or sampling domains together. And this then allows more comprehensive surveillance to simultaneously monitor the pathogens across species. And also, some of these pathogens live in the environment rather than always associated with hosts. So sampling the environment is also important. And this will then allow a more comprehensive, holistic, data-driven decision-making process to and combining appropriate contextual data. However, to get the different sectors and the different domains to work together, there's a number of barriers, and uh, these barriers uh, reduces the both the willingness but also the feasibility of intersectoral collaboration and data sharing. So the three main ones I want to highlight today are the lack of trust and privacy concerns associated with cross-domain or cross-sectoral data sharing, because we don't typically work with people in a different sector or in a different domain. But also system-wide, there is a challenge associated with the different sectors using different terminologies and languages, and also using different computing systems. So, for example, this picture here shows that in the agriculture sector, what's referred to as a plant versus in the manufacturing sector, what's referred to as a plant are two different concepts, but they're the same word. So contextual data is critical for the interpretation of the sequence data. And there are many types of contextual data. It can be used to document the sample collected, document the laboratory and the clinical processes, and it could also document the method used and the data provenance. So who owns the data and who processed the data. So it has various utilities as well. So if we want to standardize these domains of knowledge, we need to avoid a situation where if there are 14 competing different standards, then someone say, okay, let's come together and develop one universal standard that cover everyone's use cases, and then end up with yet another standard. So now there are 15 competing standards, right? So can we solve this dilemma? Can we solve this problem? So the answer is I would say yes, and or I would say it's possible, and from our own work, we are starting to see some tractions and starting to see some positive results from the approach we took. So that's what I'm here to talk about today. So we start off with defining, of course, the vocabulary that should be used, but we're not just coming up with a set of vocabularies willy-nilly based on our own understanding of our own domain. Rather, we take a much more principled approach. And this is an example of a resource that we developed called the food ontology. And it subscribed to both semantic and also syntactic standards to allow interoperability. So the design is such that it's domain agnostic and can be extended to other domains, even though currently we apply it to food. So this then allow mapping between different international standards. So we have mapped our terms, these sort of domain agnostic and sector agnostic terms to FDA's product code information and also to USDA's nutritional databases, the Food Data Central. But we also aspire to also connect it with the EU-based systems such as FoodX2. And the reason that we can do that is we essentially take a complex description and break it down into logical units that are mappable. So in this case here, organic mashed carrot baby food jarred can be broken down into parts that are related to claims that it's organic, the processing method, the type of food or the type of food product, the consumer of this food product, and also the packaging information. So each of these essentially is a independent term in food but can be combined together to describe a complex idea. And this takes away any assumptions that are built into a specific domain as much as possible. I would say right now that it's not possible to develop a set of descriptors that are completely universal, and that's why later on you'll see that the importance of working with the stakeholders directly to cover as many different use cases as possible, but also subscribing to these design principles to ensure that the terms are as generic as possible. And we actually just put up a 10 simple rule preprint about this topic. So applying this idea also to genomic epidemiology, we have a collection of terms under the umbrella of genomic epidemiology ontologies. So the aim is to integrate genomics, labs, clinical, and epidemiological data critical for whole genome-based microbial pathogen investigation. So there are different domain knowledges that we are collecting under one umbrella systematically. And actually, we're still in the process of making sure that this collection of terms is logical and so on, but this is still work in progress. But it's important to recognize that interoperability has to be engineered, and it doesn't just happen um, uh, naturally or organically. So we are part of a community of practice called the Oval Foundry. Um, and we recognize that interoperability depends on having common architecture of uh, the of how we describe data. So this is through the use of basic formal ontology and more recently some other upper level ontologies that are used to provide the foundational structure of how knowledge should be represented. And then we also, within these group of ontologies, there's also relation ontology that describes the common types of relationship between entities or between things. So, and there are some additional rules, like for example, this is an open source and open participation community, but there's some oversight needed to ensure uniform practice and principles are adhered to. And it's again a community-driven effort to do so rather than someone developing a resource completely in vacuum. So we apply this approach in the last, I would say, about 10 years now, with some interruption during the pandemic, to AMR standards. So this is a project with a number of federal departments and agencies and more than 15 universities and some agricultural environmental networks. And the goal of this project is to use genomics and harmonize contextual data to understand foodborne antimicrobial resistance in food supplies and in an environment, and identifying interventions and risk associated with these with the practice, right? So it's One Health in nature across different sectors, and it's also across different commodities, different environments, and different hosts. And then these each of these community has its own sampling strategies and sampling practice, and then using different methods, for example, for uh, um, processing their samples and tracking their own processes. So um, this is just to highlight the uh, diversities or the variations across systems that we have to deal with. Again, including different sequencing platforms or lab protocols. So using our so during this process, essentially a few of the data specifications were developed along the way. As mentioned during the pandemic, Emma Griffiths and others in my group led the development of a SARS-CoV-2 contextual data specification package for uh, um, SARS-CoV-2 genomic epidemiology. Um, but we also have applied the same approach to tracking microbes in food chain and so food production systems, and also have extended it to different pathogens. And the reason we can do that and still keep everything as compatible as possible is through the use of modular and iterable data standard design. So the idea is that we have modular framework and core content that, for example, is defined in an ISO standard, and then the modules are populated with fields and terms from community-driven specification development process. And uh, when a new use case come up, we don't just design a brand new standard. We reuse as many of the modules as possible, and then through consultation and through supporting the users to get their existing data into the standard and to test the robustness of the standard and to stay engaged with this community, we then over time have built up several different standards that are all openly and publicly available for anyone to use for free. So here are some example of the thematic modules that have been developed in the last few years. So as I mentioned, the challenge we have is not just making the data human readable, but also machine readable. So underlying all these standards is a common data modeling language called LinkML. And this LinkML modeling language allows us to bridge uh, agency-specific systems. So the by doing so, it allows us to interface the agency databases with the common standard and therefore be able to import and export data in and out of these systems. So a LinkML schema, will probably not go into the detail here, but it represents the data structure for tables, relational databases, and also knowledge graph. So it's robust enough to represent data structures of different complexity from a flat table to a database to a knowledge graph. And the schema holds a collection of one or more of these classes that may be interrelated along with shared attributes and pick lists, meaning that we define both the data fields, but we also, as much as possible, provide a list of suitable vocabularies for each of these attributes rather than just leave the users to input their own free text values in these fields. And again, the whole schema or the whole data specification is machine readable. So in order to get the users to input their data in and check if it's compatible with the data specification, we also developed a tool called the data harmonizer that enables standardized data entry and validation. So as you can see here, you can pick, there's different settings you can do, but also you can open an existing file or cut and paste an existing set of values into the table. And there's a validation button allow you to then check whether your existing data meets the standard. And if not, it gives you an opportunity to update your data. And one thing we're playing around with now is starting to use different natural language tools to see if we can do the data correction actually semi-automatically with user input, but essentially provide suggestions for users on what the closest matching standard terms is and so on. So this tool became quite a robust tool that was used by the different public health agencies when they're sharing data with the national lab during the pandemic. And we're also building in now the functionality to allow not just data entry, but also the whole data specification to be built from the same interface. And here's the example showing that because the underlying data structure is machine readable, we can easily, well, relatively easily introduce a different language and to allow the same interface to be used, in this case for French, to satisfy the two official language in Canada. Yeah, and this is just showing you the underlying data that's mapping between English and French. So over time, we have developed the entire tech stack to improve interoperability. So these are our chosen framework for the standards and data specification development. We use GitHub or similar online repository to store our code and to make the textual content available, again, open source, publicly available. And we use LinkML to describe the data in a machine readable way. We use the data harmonizer to design this data schema, but also to use for data entry and validation and conversion to different local databases. And then all this is based on based on Oval Foundry style of ontologies encoded in OWL. So this semantic layer, meaning that there's a relationship between terms, are then defined using these semantic web technologies. And this allows us to solve a number of data harmonization challenges over time. And we also, so here is just a list of domains that we that benefit from this type of approach. And we also accompanying the standards with human readable instructions. But despite having the system in place, I think it's critical to realize that human creativity is boundless and ingenious, right? So even though we designed a universal plug, a universal converter, we still have to engage the stakeholders for training and education. so they don't do something on the right-hand side here, right? Essentially reinventing the wheel, but come up with a Frankenstein plug that does work, but unnecessarily fragile and unnecessarily complex. So, as mentioned, trust is a major issue with intersectoral collaboration. Uh, so we also work with others, for example, Yanjali and Mon Dawati from McGill University to uh, look at privacy and confidentiality related issue associated with sharing pathogen genomic data. Because we know that if you combine sufficient number of fields that are not directly identifiable, but indirectly identifiable, then this could still lead to a sample being attributed to an individual. That, in other words, the individual's identity being discovered through the publication of these so-called de-identified information. So both legally, but also through public opinion, we try to understand where the sentiment of the public lies. So during the pandemic, we also conducted a survey of about 5,000 Canadians around across the nation, and essentially asked them about what type of anonymized and basically de-identified data that of COVID-19 data that they will be willing to share with researchers. So these are data collected through public health, through the healthcare system, but are they willing to have that data be available for research? And the key is that these data are collected without consent. So the question really is, uh, do they think that this de-identified data can be made available without a prior consent? And actually, what we found is quite promising, although this, of course, was done during the pandemic, that Canadians are by and large comfortable with sharing de-identified COVID-19 data for research that can be obtained through public systems and registries. So you can see here for things like symptoms, regions, geographic regions, vaccine status, hospitalization status, age, reinfection status, health outcomes, sex, and reason for COVID testing, and so on. The light green shows that these are the types of data that people are willing to share. The darker, I guess the turquoise color is that they're not sure, and the dark blue is when they say no, they don't want to share. So you can see that between 70 to 80% of the people we interviewed are okay with sharing the identified data of those types. And only when it comes to sharing their own genomes or have the data linked back to their identities when they start to show um, some hesitations, more uncertainty, as you can see here, but also some resistance of sharing that data. So collecting the data need to be contributing to positive outcomes. As I say at the beginning of this talk, there are many studies, researches that are done to show that the genomic data of the viruses combined with contextual data indeed can be very powerful for creating new knowledge. But here I want to highlight one of the tools we developed uh, to help visualize the data and therefore add value to the data, and to also highlight how this data benefit from the open science um, data that was made available. Um, so this, sorry, I should mention this just got published this year after about 5 years of work for those who are interested. And essentially, this is a tool that has a backend that calculate genetic variations across a large number of lineages in SARS-CoV-2 or other viruses. And it consists both the bioinformatics workflow, a graphic user interface to essentially highlight the mutations found in the genomes, and then linking those mutations to the known functions through literature search. So so as you can see here, this tool allow us to, if you just pay attention to this column here, to allow us to observe over time, right, from week 31 to the end of 2022 or beginning of 2023, actually, this particular mutation has increased in the virus population over time. So this is an emerging mutations, and the function of that mutation is known to affect the binding affinities to the human receptor. So therefore, this information and knowing that this mutation is on the rise can be quite valuable for epidemiologists. So this, as I mentioned, rely on manually and now semi-manually, so with some large language models data extraction support to pull function to mutation linkages into a database to support the annotations in the visualization tool. And as I mentioned, we have extended this from SARS-CoV-2 to support visualization of wastewater data and also pox data, mpox data. So as I mentioned, we benefit a lot from open data movement. So open data is a global initiative that promotes the idea that data, especially data generated by government, public institutions, and research, should be freely available for everyone to access, use, and share the data. And the goal is to increase transparency, foster innovation, and enable public participation by making data openly accessible in usable formats. So this movement supports collaboration, accountability, and evidence-based decision-making across society. So this is according to ChatGPT, right? Basically asked ChatGPT to define open data movement. And it actually came up with a very good, I would say, definition about what open data movement is. And perhaps not surprising because ChatGPT really is a product of open data movement, right? It scraped a lot of data open from open sources, but it also, of course, scraped a lot of non-open source data, but that's probably another conversation. But based on Open Data Foundation definition, open data essentially is data that can be freely used, shared, and built on by anyone, anywhere, for any purpose, right? And this is a picture showing the various social, economic, and performance benefit associated with making more data available. However, open data does not guarantee equal benefit, right? So not everyone has the technical skills, the tools, or the resources to use it effectively, which can widen inequality rather than reducing them. So there's privacy and confidentiality concerns, there's security concerns, there's economic competitiveness concerns. And then there's, of course, also risk of misinterpretation and misuse of the data that are out of context or, not interpreted properly. And there's, of course, preparation and maintenance costs associated with making the data usable by others, but that does not necessarily benefit the people who generated the data in the first place. So that's really all these reasons explain why more people are interested in generating data for their own use and then put out low quality data in in open data repositories rather than spend the time and energy building high quality data because they can't use other people's data, so why should they make their own data more reusable, right? So there's quality and reliability issues associated with open data. And it also highlighting the care principle or the failure to meet the care principle where create these principles created to ensure that data practice respect indigenous rights, promote collective benefits, and uphold ethical responsibilities. In other words, to bring the human and cultural dimension and considerations into the data governance practice. And this, of course, has some underlying, these principles are based on some underlying practice and underlying documents, such as the UN DRIP, the United Nations Declaration on the Rights of Indigenous Peoples, and to reaffirm their data sovereignty. So So we know that there is really no one-size-fits-all framework for data governance, and the data governance need to consider organizations' own needs, and the data frameworks need to be put in place to address things like, the use of data in supporting program goals, the data standards used, policy, and the process used in the organization. So we're not trying to create one standard that replaces everyone's standard, but rather to trying to create an ecosystem where interoperable standards or practice can come together, and then the differences will be can be resolved because they will be minor and they'll be based on common set of principles and practices. So therefore, just to end with the idea of maybe we need a new norm for pathogen genomic data sharing and move away from expecting that all sequence data to be deposited in open and public repository to come with a minimum contextual data, which often result in large amount of sequence data with low quality and missing contextual data. Instead, we should focus on building interconnected, interoperable data repositories with flexible data governance, respecting the data sovereignty and sharing and the shared benefit, and with clear defined data access and use policy. However, that's not the end, right? We should be thinking about when the data over time becomes less sensitive, and or if the case number increase to the point that re-identification will be much more challenged, then there should be a pathway to allow more and more de-identified data to be deposited into open and public repositories with high quality sequence and high quality contextual data. So to end, we propose this ecosystem approach based on three principles. And actually, some of you may know this approach. We propose it for an agri-food system, actually, but it's equally applicable here in public health. So the ecosystem should be open and inclusive, and encourage partnerships from diverse domains and sectors. And then it should be open, based on open technology stack and community developed standards for the core component of the ecosystem, right? So that mapping standard should be community developed and open, but people, of course, are free to uh, create their own local standards that are compatible and mappable. And then the data governance should emphasize on equitable data sharing and collective benefit sharing. So in summary, pathogen genomic data provides rich information and can act as common currency across the sectors. However, the lack of consistent contextual data can prevent aggregation of pathogen genomic data for research and analysis. So interoperability need to be engineered by building fit-for-purpose modular data standards based on sound semantic and syntactic principles and the tools to enable mapping across local specifications to standards. We and others have developed tools to enable large-scale visualization and analysis of genomic data to extract biological and epidemiological insights to show the values of these tools. So creating an equitable and robust global data sharing ecosystem for pathogen genomics with de-identified contextual data, I believe is within reach technically, but then improving the trust across domains is important, and therefore an open ecosystem is not a requirement to make your data open by default. Rather, it's a process of opening your data to more and more utilities. and share the common benefits of such practice. So we need to work together to develop trust across sectors and domains so pathogen genomic data can be used for research and innovation. So I'll end here by thanks a lot of people, both in my group and collaborators, and of course the various funding agencies over the years that have provided us the support, of course, including Genome BC and Genome Canada, for supporting our work. Thank you very much.
Thank you very much for running us through the different efforts that you've led or collaborated on data sharing, on tool creation, on standards and everything. It's quite an amazing. Thank you so much. I didn't know about half of the work that you've been doing, so I'm very impressed. I'll go to some of the questions that we have on the Q&A section. So compared to before COVID, how well prepared are we for the next pandemic? And maybe related to that is the next question, if the system is able to predict where the next pathogen of concern will be coming from.
I don't think the system is able to predict where it will be coming from, but it will certainly, hopefully, be able to pick up the signals faster now. And because and even compared to the SARS and SARS-CoV-2, the speed of information and travel and the speed of generating sequence data went from months to days, right? So in the first SARS, the data wasn't released for 3 or 4 months after the case was reported, and contrast that to SARS-CoV-2, it was released about a week after the case was officially reported, right? So, and the detection certainly is faster, but the release of sequence also allow global response to be initiated much quicker in terms of designing diagnostic tests and so on. And back to the point about whether we're better prepared now, I would say if the pandemic happens tomorrow, then yes, we're definitely much more prepared now. But if the pandemic happens 10 years from now, right, then that's much harder to say because I think, as we've seen and witnessed, peoples are quick to forget about the hardship, and the lesson learned often doesn't end up getting proper support by by government fundings to be put in place. So that, yeah, that's my answer. I think if, and we don't think pandemic will happen tomorrow, but certainly it will happen again. And the question really is how can we be prepared without making it cause an arm and a leg between now and the next pandemic?
Yeah, very good point. Sometimes we do forget out of necessity or self-preservation the hardship, and then the efforts are no longer sort of focused in areas where we know they will be needed at some point. So let's shift focus to some of the AI-related questions. So you mentioned that you're starting to experiment with using natural language tools for data entry. And that was, I think, specifically in your data harmonizer. Have you explored other applications of AI in your work? And how do you evaluate the trade-offs, such as reducing user workload versus the risk of introducing errors and bias?
Yeah, so yes, for sure. Like, for example, in the meeting, I use a quote from ChatGPT verbatim on what open data science is, right? So I do use ChatGPT, but also just yesterday when I asked it, how many asked me to give me a report on one of the papers, which I read when I realized the summary it gave me actually has a wrong number in it. And then I asked ChatGPT to realize that number was wrong, and then it actually responds, say, yes, thanks for pointing that out. This is the correct number. So it's actually has the ability to learn now, but it's also for sure continuing to make mistakes. So ultimately, I think the individual using these AIs need to be responsible in verifying the answers themselves. I would treat AI as as a assistant that retrieves information and things for you. But if you're going to use that information, you're ultimately responsible for that, for saying that, for using that information, right? So if I quoted the wrong number because ChatGPT gave me that number, the blame is on me and not on ChatGPT, right? But then ChatGPT can be very helpful in pointing you to pointing you in the right directions, and not just ChatGPT. Any AI assist tools can do that. Some are more prone to make mistakes. Some others are less prone, but for sure they all make mistakes.
So we have 1 more minute, but I'll try to maybe get through a couple of questions.
Sure.
When you introduced ontologies, you provided an example, and usually ontologies define atomic concepts as terms, but your example defined things like carrot root, whole, raw, instead of using separate properties to describe the carrot. Given the vast number of crops and the additional attributes, it leads to an explosion of terms, as more conditions are added. Are you aware of the specific reason in this case?
Yeah, that's a very good question. Thank you, David. The the reason is because this is a very common combination of terms, and then by having it directly in the ontology, it gets used by different users. So we try to strike a balance between that sort of combinatorial explosion of terms that you eventually have to say, carrot jarred, green bean jarred, or peas jarred, and so on. But it's a bit of balance, but all the base terms are still there, and we still encourage people to use a faceted approach. But then from time to time, if you keep doing that, at least right now during a transition, a lot of systems cannot handle that on the fly sort of composing of terms. So we end up pre-composed terms that are commonly used to support some of our users. Does that hopefully answer your question?
Okay, one last question from Daniel. Did your work on One Health data change with the global expansion of One Health to include environmental health? And were there any particular challenges?
Yes. So my definition of One Health indeed will include environmental health in the sense that infectious disease exists in the environment, as I mentioned, but also in a very important sense that infectious disease, just like pathogens, just like are subject to the effect of climate change, subject to the effect of uh, the environmental change in general, right? So contamination or so on and so forth. So climate change, for example, is a big reason why we're seeing shift in patterns of transmission and where infectious disease, where are the hot spots of emerging infectious diseases, and these are all tied to climate change. So the infectious disease that are both that the pathogens that are found in the environment and also pathogens that are associated hosts, but in a situation where there's climate change, both I would consider that as sort of environmental impact on infectious disease. So tracking the environmental variables becomes very important in interpreting these changes.
Thank you so much, Will. We're a bit over the time limit. Really appreciate your giving us an overview of some of the work that you've done in the infectious disease space. Thank you so much. Have a
You're
good
welcome.
rest of your day, everyone, you included, Will. Thank you.
Thank you. Bye for now.